All right, so we'd like to introduce Rose Leeger from, where are you from, Rose?
So I'm a junior at NOAA Southeastern University. I'm actually triple majoring in marine biology, environmental science, and biology, but I'm also a NOAA undergraduate scholar, class of 2021. And this summer, I had the privilege of being the Office of Aquaculture summer intern. So my project was on oyster probiotics, and it specifically highlighted how a bacteria found in yogurt helped boost oyster farming. And there I am in the milkbread lab handling the probiotic. So for my presentation today, I will give a little background on just the life cycle of oysters and a quick overview on Office of Aquaculture and what we do. I will then provide a little insight on probiotic research being done at the milkbread lab and some of the interviews I conducted that was highlighted in my featured story. And then I will conclude with acknowledgements and any questions you may have. So the oysters you once eat at restaurants once look like what you see on their left. They're small, microscopic larvae that are very hard to distinguish them from the algae food they eat. Before long, they settle down on solid surfaces where they attach for the rest of their lives. Now, a crucial component of global ocean health are the oysters. They are filter feeders, they help provide jobs and habitat, and they can serve as barriers for local storms and tides. They also help provide erosion and are essential for helping protect our estuarine waters. Bivalves are ocean's filters, just like the filters that you have in our fish tank, except that they filter 50 gallons of water a day. They remove excess nutrients and other pollutants from coastal waters. NOAA has hatcheries that seed oysters in areas where oyster reproduction is non-existent or unreliable. This will help establish new reefs and improve local water quality. But how did these oysters come to be? Through aquaculture. Sound familiar? It's like agriculture, but in the water. This is a huge industry, yet the United States ranks 17th in the world, which is lower than the desert nation of Egypt. This is one of the fastest growing forms of food production, with over half the world's seafood coming from aquaculture. We are the second largest consumer of seafood after China, and yet over 70% of our seafood is imported. Oyster aquaculture is the most valuable sector of marine aquaculture and is a great example of a low carbon footprint protein. Oysters are the most cultivated shellfish by value, and recent numbers show that more money was put into oysters compared to any other species. Now, oyster larvae are grown in hatcheries such as VIMS, the Virginia Institute of Marine Science. They have large tanks inside with a pipe that pumps up fresh water. They can control the conditions such as salinity and pH within the hatchery, and that helps control reproduction. Now, usually oysters in hatcheries have water temperature signals to show when they're ready to reproduce, and since they're in a controlled tank and area, this will lead to fertilization. The microscopic larvae in hatcheries can be sold, or farmers can wait a little bit longer and wait for the desired spat that they want. Farmers can then bring the juvenile oysters to the farm, which is usually off the coast, that can then be made of oyster cages or naturally made reefs. The oysters then grow till they reach the desired grade or size and are then ready to be sold and eaten. One large challenge is a bacterial disease. Vibrio diseases largely affect the juvenile and larval stages of oysters, which is their most vulnerable stage. So NOAA has a role with aquaculture all around the country, with each region focusing on different aquaculture issues. This allows for national and even global reference. For example, the Northwest focuses on native species enhancement, the Southwest focuses on fish genetics, and the Northeast Fisheries Science Center focuses on shellfish health. As part of the Northeast Fisheries Science Center, the Milford Lab in Connecticut has found a novel probiotic from the gut of oysters, OY15. This is a safe, non-antibiotic method to boost oyster larvae survival 20 to 35%, and they found larger growth as well. This could yield to longer growing seasons and help make oysters less susceptible to disease outbreaks, which will lead to less hatchery collapse. So how did this come to be? Well, it started out in 2011 with initial research done by Dr. Diane Capernico and Dr. Gary Whitford at the Milford Lab. There, they isolated bacteria from the oyster guts, and they found 26 different colonies, with OY15 having the largest zone of inhibition. They then tested it against a known pathogen, B183, and they saw immune stimulation. They partnered with other research labs and universities to calculate gut transit time, which was just how long the probiotic lasted in the oyster. And they saw that it was out in 2 days. That's because oysters are filter feeders. They then used molecular technologies such as pathogenicity genes in OY15 to prove that it was a benign strain of bacteria, so it wasn't a pathogen. Other collaborations tested for the immune system function within the oyster. They put fluorescent dye within, and they also tested with human epithelial cells and proved that it was safe for humans. They then created a public-private partnership with a company called Prospective Research, and this allowed for trials to begin. Currently, we're hoping to spread the word and the success of OY15, and we're waiting until about August or September of this year for numbers to come back, so commercialization steps can begin. So part of the steps to commercialization of a product include testing different formulations. Prospective Research created a freeze-dried formulation of OY15, which was used in a large-scale hatchery trial. To ensure that the data and research is up to date, a 3-week-long larvae challenge in June of 2021 was conducted in the Milford lab. I was fortunate enough to go up there for a visit and help out with any water changes and the setup and running of the trial. We tested the probiotic alone, the probiotic and the pathogen alone, and a control. I helped change water, and we used a sprayer net to wash and clean the larvae. We also added OY15 every other day, and this was calculated by the gut transit time. And we fed them 2 different types of algae, with the dosing changing each day as the larvae grew. We looked at the larvae under a microscope, and we saw structures and features grow and develop. I even got to see the eye spot the last day I was there. We calculated the survival rate, and this was awesome because it gave me firsthand experience to research and microbiology techniques, and I got to see the research aspect come to life, but so what? Research coming to life is always awesome to see, but its important aspect is also sharing what you found. If it weren't for the communication tools today, OY15 would not be the success of what it is. In the past, it allowed for initial internal aquaculture funding for it to grow and to be replenished in the following years for more research to be done. Now, it allowed for a cooperative research and development agreement, or a CRADA, to be formed with Prospective Research and NOAA. Since Prospective Research knew hatcheries all around the United States and international, it gave us good connections and collaborations with farmers firsthand. In the future, there's so much potential once commercialization happens and success is highlighted, it can lead to so many more uses. So here's just a few examples of the communication tools involved with OY15. Oysters are a good example of restorative aquaculture. That's well-managed commercial aquaculture to provide ecosystem services. A great example of this is the SOAR program, which is supporting oyster aquaculture and restoration program with NOAA and the Nature Conservatory. This allows for economic stimulation for oyster farmers, and it helps put the oysters that were farmed back into local habitats. Part of my involvement this summer, besides the trip to the Milford lab, was creating a featured story. This highlighted 2 unique perspectives in the aquaculture field, and it shared with the public, other companies, and other hatcheries the success and use of OY15. So my featured story interviewed Iarla Connolly, who is an Irish hatchery farmer who runs one of 3 hatcheries in all of Ireland, and Dakota Hamel, the CEO of Prospective Research, which was a private aquaculture research company. Iarla had just finished a trial using OY15 in his juvenile oysters, and he explained that June was a problematic month with larvae growth, and since antibiotics cannot be used in shellfish, he was looking for alternative methods. He ran small trials and found huge success and would love to run more. He explained how his hatchery showed the symbiotic relationship between research and real-world applications. It's now not something that in a beaker, there's real-world effects seen. Dakota explains the research to commercialization steps and how he saw how probiotics, a direct-fed microbial, were the perfect food supplement. He's hoping to create the perfect one that can allow to help increase the success of shellfish farms. And his company is trying to figure out the easiest way to administer the probiotic to hatcheries, as different ones have different sized tanks, so it could affect dilution. Without a doubt, he'd love to use this internationally. It would be awesome to see if it helped with Ireland's native oyster population. And in the picture in the middle, the bottom one is the batch of OY15, and he explained how it's being fermented and then being put in a pilot scale lyophilizer which is then freeze-dried into a tray, and then they grind and mill the powder and dilute it into an inert carrier. In the future, they're going to be packaging up the next round of OY15 before spawning season and putting it up on their website. So here are the two featured stories I was able to be part of during my summer internship. These two stories overall got hundreds of views just based on our communication specialist when first published. The OY15 story was featured under Milford Lab in the news in the NOAA Milford Lab website. It also was featured under Florida Aquaculture Association homepage and even 6 coolest Northeast aquaculture science collaborations to follow. The other featured story documented my time and memories as an Office of Aquaculture summer intern, along with the other intern at the office, Benjamin Hurley. Overall, the success and news of OY15 expands far past the Mid-Atlantic region. Numerous hatchery trials are happening in Washington, Hawaii, Massachusetts, and even the one in Ireland. This small-scale research that started out in Milford only 10 years ago now has global growth and trajectory. It has benefits worldwide for shellfish farming industry. It provides coastal jobs and ecosystem benefits. Aquaculture is expanding on a worldwide scale. Wild fisheries alone cannot meet the increasing demand for seafood. This is a game changer for the long-term perspective of the world. We're struggling on so many issues globally, such as food shortages, but we have research and science to combat it. OY15 is ready to market, and the research says it's a go. It increases oyster larvae survival 20 to 35%, and we're saving the oceans. This is the kind of research on how we save the world. OY15 is impactful and transformative into making global aquaculture more sustainable, and it's just one small step in helping aid and create sustainable food for a growing population. If you scan this QR code with your phone, it'll take you to a link to the featured story I published with NOAA last year. I'd like to thank all my mentors, specifically Steph Bierkoff, who was my main mentor with the Office of Aquaculture, along with Diane, Gary, and Kristen from the Milford Lab, and the Office of Education for seeing my potential and allowing me to share this story. Here's my contact info. If you notice, I'll be in La Jolla, California next summer, and I'll actually be in a totally different section of oysters and aquaculture, helping with their Antarctic field research and helping train their AI to look at satellite penguin populations. And thank you very much. You guys
A few minutes for questions. Dr. Shah.
So it's really nice, beautiful presentation, thank you. And I love the history. I just want to say for you and for everybody here that back in 2010 when that started, it started with an LMR CSC intern sequencing the 16S genes of OY15 at UMSI. So Amar Hanif is now at NOAA headquarters working on harmful algal blooms. He got his PhD through UMSI. So I just love how the LMR CSC has been, the EPP has been involved in this from the start to now. Really got going.
Just, sorry, I missed that part. Maybe you apply probiotics with algae, right? It's together and it's applied to the oyster larvae?
Yeah, so the algae is the food they eat, and the probiotics was the immune stimulation that helped them boost and grow.
It was applied together. And how old was the larvae when you apply? Is it like how many days old?
They were only like less than 2 weeks old. The 3-week larvae challenge was from the beginning when they were spat and they weren't even out of bleeding from. And by the end, we could see them on the beakers and on the cups, but it was a 3-week long. So from when they were first hatched to about when you could see them without using a microscope.
Good, very good. Thank you.
Do we know whether these probiotics work with all kinds of species of oysters, or we don't know?
That's actually one of my first questions I asked Diane when I went to the lab. And she said that they're doing some things with the Pacific oyster, but we can't bring the Pacific oyster to the Mid-Atlantic because of contamination and different salt waters. They're hoping that they do it with the Pacific oyster, but I actually think that their next step is to use it in shrimp. They're either going to extract the probiotic from the adult shrimp or even use OY15 within shrimp, because someone did like a preliminary research and just adding the OY15 in shrimp saw boosted larvae survival as well.
It does work with the gygos.
Okay.
Yes. Yeah, I know because I was in the pet.
Quite a few questions, but because I'm coming from a perspective that I don't must know much about oyster farming or hatcheries. So after you inoculate these oysters with the probiotic, what type of environment will you be containing them in so they can control it?
Right, so the probiotic is only administered when they're in the larvae stage, the bacteria stage. And then once they grow and they've been administered that, they just go to regular hatcheries and do regular types of aquaculture. So it's just the beginning phase, which is why it's really important to have the connections with the hatcheries, especially the larval stage of them. Since there's a lot of oyster hatcheries that already have them once they're large and they've been extending on things, but this is like specifically for just this life stage, because that's when they're like most vulnerable for filter feeding and administering the pathogen and disease.
Okay, the second part of the question is, okay, now that they are being allowed to mature in their environment, what is being done to address, is the probiotic helpful or beneficial in assisting the oysters become more resistant to climate change and the implications of it?
So ideally, that's the goal, because why do all this if it's not going to help the future? Specifically right now, E183, which is the main pathogen we tested against, is mostly found in diseases not related to just climate change. It's just a bigger disease that happens and causes a lot of hatchery outbreaks and diseases in falls. Of course, that is related to climate change, because as climate change gets worse, there's more diseases out there, there's more pathogens that can affect oysters. So definitely well affected, but specifically D183 is not directly related to climate change.
Understood.
All right, that's all the time we have for questions. Let's thank Will.